All right, welcome. We are live today from Vienna for a very special episode of the Immunology Podcast. I'm Dr. Jason Goldsmith. This is Dr. Brenda Roud, and we are here live from IUIS 2025 with a special live episode in front of a live audience. We're going to have a great conversation today talking about mRNA-based dendritic vaccines for both liquid hematologic tumors and solid tumors. And with that, I'll let Brenda introduce our guest here.
Hi, everyone. Well, so excited to be here. And we have our very special guest with us. We have here with us Professor Zwi Berneman from the University of Antwerp. Professor Berneman is Emeritus Professor of Hematology at the University Hospital of Antwerp and also former head of the Department of Hematology over there. And Dr. Berneman has extensive experience developing dendritic vaccines for cancer and also tolerogenic dendritic cells for autoimmunity. And we're going to be talking to him today about 20 years of dendritic cell vaccination and his experience in this very important field. On top of his, moreover, Dr. Berneman is co-medical director and helped brought into being the Institute for Cell Therapy and Regenerative Medicine, also at the University Hospital of Antwerp. And we're very excited to have you today, Professor Berneman. Thank you so much for joining us.
Thank you very much for having me.
All right. So to start this off, we're going to kind of go on the journey of the development of what you've been working on. But to start, can you provide a little information what a dendritic cell vaccine is versus, my standard flu shot? And also, where are we at clinically today? Where do we stand in terms of it being on the market for patients?
Well, dendritic cells are the conductors of the immune system. They can stimulate the immune system, for instance, the T lymphocytes, to go on the attack against virally infected cells or against cancer cells. But they can also have a tolerogenic mode, and they can dampen the T cell responses. And so if you use those dendritic cells, you can use immunogenic dendritic cells to treat cancers, to help treat cancers. You can also use tolerogenic dendritic cells to dampen autoimmune diseases. Now, usually, dendritic cell vaccination, like it is called, is used in the therapeutic mode, so when the patients have already a disease. But we've heard at a conference from Jolanda de Vries, with whom we've collaborated, who has helped us actually set up our whole system in Antwerp at the time. She's from Nijmegen in the Netherlands. We've heard from her about preventive dendritic cell vaccination in patients at risk of developing cancer. So this is the basic setup of dendritic cell vaccination. Now, for the dendritic cells to be active, you need two things. They have to present an antigen. So you have to, because you want them to have a specific effect or almost specific effect. And there are different ways to load antigens in the dendritic cells. And the second thing is that they have to have an effect on so they present these antigens to the T lymphocytes. And then they also have to have an effect on the T lymphocytes. In the case of immunogenic dendritic cells, they have to stimulate the T lymphocytes. In the case of the tolerogenic dendritic cells, they have to dampen the activity of the T lymphocytes. So those are the main hallmarks of dendritic cell vaccination. When they're used in cancer, they are part of what we call the cancer vaccination, so treating patients who already have cancer with specific molecules or specific substances which are also present in the cancer cells and against which immune response should be stimulated, because they are really the orchestrators, the conductors of the immune system.
So you started working, your first clinical trial started 20 years ago in 2005. And it feels like such a long time. But I guess immunology was a little bit different back then. So we've learned a lot. But why don't you tell us a little bit about the motivation and what was the state of the art around that time when you started that motivated you to go down this road that has taken you this far?
Well, it was already, it existed already, and dendritic cell vaccination at the time. Also for other forms of cancer vaccination, it got not such a good rep in the field because usually the level of overall responses, being partial responses or complete responses, is relatively low, around 10 to 20% of the cases. And so the classical oncologists want to have, when they use chemotherapy or an agent, they want to see a response of at least 30% reduction of the tumor itself. What has become clear, and I put this parenthesis, but it's an important parenthesis. It's true that it's still in our hands. When it's used alone, it has a low level of overall response. But what has become apparent for immunotherapy in general, not only dendritic cell vaccination, but also the immune checkpoint inhibitors, is that in many cases you don't have an overall response, but you have stable disease and you have lengthening of the survival. And this was not calculated in the beginning, when many trials were being done, and so this was already, and it has become a more important feature nowadays, to try to incalculate that and to look at overall survival in order to be able to judge that. So as far as we were concerned, we started working. Actually, when I came back from America in 1993 to take over the department, I was stimulated by my former boss in America to work on gene therapy. And so the first idea was to develop non-viral methods of gene of transfection because of operational reasons, because of safety reasons, and to do that in the stem cells. But very rapidly, we went on to culture dendritic cells. We published different of those things. And then we were thinking of methods to load the dendritic cells, like we have done for the progenitor cells, the human hematopoietic progenitor cells. And we were working with DNA electroporation at the time. We thought it was working. But in the dendritic cells, when we used DNA encoding tumor-associated antigens, there was a stimulation, but it was not specific. So you could not see if it was specific. Nowadays, we understand why, because there are immunogenic sequences in the DNA which will stimulate the effect. So you don't know if the effect is specific or not. Yeah? And then by the late 90s came the seminal publication by Ellie Gilboa, the group of Ellie Gilboa at Duke University, about pulsing dendritic cells with naked mRNA, unprocessed, so unmodified mRNA. And he described that was in the context of carcinoembryonic antigen, where they saw an effect. And they did some clinical trials in Duke. And then my collaborator, Viggo van Tendelo, switched to mRNA and instead of using DNA, but mRNA, encoding. Yeah?
Were people surprised that you could just pulse mRNA and it would work?
I think it was surprised. Yes, it was surprised. And I was told that the FDA also was having a question mark about that, And when our data came out of an increased transfection efficiency with electroporation, then probably they started believing it, And in the meantime, the RNA electroporation technique has become a very widespread technique, not only in the field of immunotherapy, but the CRISPR-Cas system is using very often electroporation. So it's this has been one of our contribution. This was 25 years ago, where we showed that the electroporation technique was more efficient, both in terms of transfection efficiency as in function of the T cell stimulatory capacity, the specific T cell stimulatory capacity, then passive pulsing of mRNA, and then lipid encapsulation of the messenger RNA. We're still talking about unmodified RNA. Yeah? And so then we had already something in our hands. Later we also published mRNA electroporation in T lymphocytes and in stem cells, so in human hematopoietic progenitor cells, also in embryonic stem cells. That was in 2004. But we concentrated our work on the on the dendritic cells. And then we still needed a confirmation that the method, we used to work with melan-A in the publication that we published in 2001, melan-A or MART1. But I'm an hematologist, and it's not a very strong tumor-associated antigen. Plus, I wanted to use something very broad, with a broad activity. And I went to a
melan-A is mostly in melanoma,
melanoma,
right?
yes. And even in melanoma, it's not considered to be the strongest antigen, But it was a proof of concept. We had a proof of concept in vitro. So I heard a talk by Hans Stoss from London at the Vilseder conference in Germany, showing his effects in mice with T-cell lines against WT1, the Wilms tumor antigen. And he introduced me to the field of these nearly universal tumor-associated antigens, which was pioneered by the group of Haruo Sugiyama in Osaka, Japan, with whom we've been collaborating, and also the group of Seattle from Phil Greenberg. They were working on WT1. And then I said to myself, okay, we have our antigen. This is what we're going to use. And it's also overexpressed in the majority of the tumors, So we had something that we could use in different tumors, which was also our intention. And then we went to Hans Stoss' laboratory to expose our WT1 messenger RNA electroporated dendritic cells to his T-cell line to see if there was a specific effect. And lo and behold, there was a specific effect. And then we decided we have to go into patients.
Is that the GP100 cell line? The T-cell line against WT1?
It was an allogeneic restricted cell line, from which they extracted later on the T-cell receptor, So it was actually, and this was a moment, okay, then we decided, okay, we have to go into patients. Since we are hematologists, we are still faced in acute myeloid leukemia with a very poor survival. And then this is the way we started the trial. And we started seeing effects, when especially on the minimal residual disease tumor marker levels. And these effects were correlated with the T-cell effects against the WT1. And then we started thinking together with our colleagues, the oncologists, the solid organ oncologists, let's also try to tackle other very bad cancers where there is a high medical need, And we already had evidence from the group of Japan. In Japan, they are doing peptide vaccination, and they had evidence for activity in glioblastoma multiforme, for instance. So we said, okay, let's go for that, and breast cancer, metastatic breast cancer, and in mesothelioma. And mesothelioma is also interesting because there is a high level of WT1 expression in the tumoral cells there. So this is the way I'm thinking about the review I read in the early 90s from George Klein, one of the fathers of immunotherapy, who the title of this review in the annual review of immunology was How One Thing Led to Another, And so this is, I'm thinking when looking back in retrospect. And he ended this abstract, we have only scratched the surface. And listening to everything at this conference, I think he was deeply right and is still right to this day.
So I think I'm picking up that when you do a dendritic cell vaccine, are you going to take dendritic cells from the patient and then electroporate them or do something ex vivo and then place them back? Is that the approach, or what are we doing today for dendritic cell vaccines? Well, there are different ways
to do it, and the GMP aspects and the production aspect is also playing a role. The way we've been doing it is the classical way of culturing them from peripheral blood monocytes, because then you can get a certain amount of cells. It's still a pharmacological concept to try to get enough cells, to override the immunosuppression which is in the body, So you need to, according to some concepts, you need to have a certain amount of cells. So we're culturing them from peripheral blood. And then afterwards, after the culturing, which takes 6 days, when we started it, this is another issue. When we started it, the only thing we had to conform to were the laws of the tissue banking, which say that you have to make sure that you identify the donor. You have to make sure that if you're giving it back to someone, it's the same person. You just to document everything, to have standard operating procedures. But then in 2012 came the European directive saying that genetically engineered cells or cultured cells had to be considered as advanced therapy medicinal product, so as to be considered as drugs and have to conform completely without exception to all the regulation of the pharmaceutical regulation. So we had to start at the time, and fortunately we were not faced with that in the beginning. But then we had to decide, we have to conform to the law. There's no way to escape that. So we had to, the costs were increasing. We have had to have more stringent clean rooms to use that. And in that we were helped by the hospital, by the institution, and by the grants that we received, but mainly by our institution. And for instance, you need to have a qualified person, so an industrial pharmacist who is releasing the product. So I was responsible for the product, but I was not allowed to release the products alone because I cannot be a controller myself. So it has to be the person, and so that person is costing €1,000 a day. But it was a good investment. I mean, and there's no question whether yes or no. It is compulsory, and that person has to be very high up in the hierarchically scheme of what you have to use. So this is coming back to the question. So it made the things more complicated, the paperwork more complicated, more work, more costly. And so some people nowadays are they extract the dendritic cells right away from the blood. And sometimes they even not culturing them, So then they are not even considered to be ATMPs. So then it's a little bit like stem cell transplantation, that it's not responding to these stringent rules, And sometimes they are modifying them, those dendritic cells, just by overnight culture, so it's not such a long procedure, So this is dependent on the availability of resources, it all costs money, and all the money that we have put in that was given to us by our institution and by charities and by grants, competitive grants, which we've had to. So there are different ways of using dendritic cells, but the most common way is still I think for culturing them, because you can get a large amount of cells in this way.
Yeah, I guess that coming back to the topic of how different it is from an idea in the lab, and you culture the dendritic cells, you make your monocyte, you make your dendritic cells, you load them, in vitro works great in a mouse, and then it comes to bring it to patients. And you already mentioned the challenges of not only making them at the right amount, so getting enough product that will be clinically meaningful, but now and increasingly more complying with all the regulations and the standards that ATMP production, so these advanced therapeutic medicinal products, they have to comply. And that makes it very hard, right, for academic centers to really continue this kind of work, and you have to be very strategic on what you are doing. So I guess, what has been thinking in the mind of an academic center and the production of these therapies? You already mentioned, for example, increases in the quality control and those. But what have been the milestones that you have had, that you now have a GMP production facility at Antwerp University, and several other academic centers have this kind of infrastructure? But what have been the steps to get here that were most meaningful, and you think that every academic institution has to go through?
It's a complex question, but I think first you have to have a department that is ready to invest the time, the money, the personnel. And to have interest in those things, and to have a certain continuity of that, And so this is one thing that is absolutely necessary. Without that willingness, it's not possible, Then you need to have the personnel is a very important matter, And where we have been blessed too is that different people who have worked as basic researchers in the laboratory, following their doctorate with us, they have remained with us, and they have converted part of their activity into the cell therapy field. And they have also added knowledge in the regulatory field. This is something that we've had to add. So the personnel that you need, and the doctoral fellows are wonderful, but after a while they leave, and so to keep the personnel and to keep them interested and to keep money for them in an interesting way is, I think, a very important thing, and so we have been rather successful. Then we need to write grants, and I think our success was based not only on the research, but mainly on being able to write grants so that we could do the clinical trials. And in Belgium, especially in the northern part of Belgium, there are different, at least one charity which is really raising a lot of money for research, and they are interested in sponsoring trials in patients that are not supported by companies, by pharmaceutical companies. So the high then you have to identify the unmet medical need, because you're not going to do a Me Too trial. The Me Too trial with known compounds, this is then sponsored by pharmaceutical companies. So you have to have this setup. It started with the willingness. It started with the technology in the laboratory, then with the willingness of embarking into the clinical trial, using that, developing the whole program, not being burdened with these very strong regulatory issues in the beginning. Yeah. So, which made us think, okay, are we going to abandon it, or are we going to continue and just to add the regulatory burden? And so we decided, okay, we want to continue. And what is also helpful is if you have some success, then it helps you advance there. I put a lot of strength in. I got a lot of strength from my past as a hematologist, because in hematology we are used to cell therapy, which is not in the hands of companies. I'm not saying it was not at the time in the hands of companies. So stem cell transplantation, bone marrow transplantation was in our hands, and we had the freedom to operate, but we had to, of course, to conform to the rules. But this is something that we were used to, So it was not completely strange for us, to go into the cell therapy field. And we have a freedom. If you conform to the rules, you can have everything in your hands to operate. You're not dependent on company. And even with time, in the beginning, we ordered our mRNA from companies, but the prices have increased. The availability of production has decreased. So we've finished up since a few years of producing the mRNA ourselves. So these were the different. I don't know if I responded clearly to your answer, but the different steps, And I'm always thinking about Rita Levi-Montalcini, who got a Nobel Prize for the growth factors. She was asked what she said that in her mind, the success in research did not depend on how intelligent you were or how much you knew, but in underestimating the problems you were tackling. And I was, but thinking back, I think there's a lot of truth in it, But you have to be determined, that you want to go for it. Yeah?
So to your point, GMP is really expensive, and once you have it, you have to maintain it, right? You can't just turn it on for a trial and turn it off. So you acquire all of this overhead. And, that's one thing in industry where you have like 5 or 6 products, you have money coming in all the time, and you can do that. But when you work at an academic institution, you're on grants. So how have you guys been able to navigate that space of, I have a GMP facility, because that 1,000 euro person day, he's there 365, or she is there 365 days a year, regardless if you have a trial going on or not. You can't just get rid of them on and off. Essentially, it's not a switch. And so how are you doing the funding to be successful in these institutions when you're running on grants?
Well, for the moment, the hospital has supported us from the beginning. So they established, once we started doing the trials, they established the cell therapy facility, which was first located in the hospital and then later on in a separate spin-off. But even in the spin-off, we pay for the technicians and for the reagents for our cell therapy products that we use in the trial, And this we get from the grants. different grants from the charities, from government-associated scientific organizations. And then has been also a foundation linked to the hospital, which was created then. And then I convinced them also to start a campaign. So it's they are raising money from wealthy individuals, but also from small donors, But so there's a campaign regularly, But it's a permanent effort, So the role I had, and which my successor, who developed certain of the techniques I talked about, and I was also branched off in the CAR T field. This is 4 or 5 full-time jobs, that you have to tackle then, But if you need a little bit of motivation there, and especially the young people we've been associated with have given us a lot of energy to be able to continue, So,
yes, for youth.
What's that like, Brenda?
But it's a very good question. So it has to be sustained. It's not an easy effort. The grants run also out. So we started also going to the government to ask them, because we were told by people, okay, you have to get then you have to get a marketing authorization. You have to put this, but this is costing money. You have the recurrent costs, and it doesn't even guarantee you in the end that you will get reimbursement of these cells. So what we've done, and what we're still going to put some effort in, is going to the government and ask them to reimburse the production of the cell therapy based on the studies where we have shown some clinical activity. And there we are encouraged a little bit by what we have had with the stem cell transplantation field, where it took I think we did our first stem cell transplantation in our hospital in 1983, and I think it lasted 20 or 25 years before the government started reimbursing. The patients were when they were in the hospital for the transplantation, the costs were paid for it, but the hospital or the government didn't pay for quality control, didn't pay for all those things. So we had to carry that within the budget of the department, which was then highly negative, So it took 25 years before the government started reimbursement of certain activities. And so I hope, at least for certain cell therapy products, that we will be able to convince them. And we were in negotiations, and then came the crisis, the COVID crisis came there, and then the whole priorities changed for a while, And so,
I mean, that's another aspect, especially here in Europe, where you have things, what gets reimbursed is usually the government has to say. At least, for example, in Holland, where I live, the insurance companies have to cover according to regulations done by the government. So it's a constant negotiation. For example, like there you have clinical trials, also cell therapy, TIL therapy, and it was only possible thanks to this kind of indirect support through the reimbursements of the health the health agencies. And I think that's probably the case in many places in Europe, at least, where we have this arrangements with government. But that's also a very important aspect. And maybe you said, I'm just a hematologist, and now I have to go and negotiate on behalf of this.
It's part of the job.
It's part of the job, right?
So this is... especially if you feel that you have something that with some clinical activity and which is still responding to unmet medical needs, There has been a development in the field of cell therapy with the CAR T lymphocytes, which and fortunately certain companies have gone into the field, and have convinced the government for reimbursement. It's a high cost, but it's for instance, for multiple myeloma, which is where the costs, I think I'm not sure anymore, but there are very high costs with the drugs in multiple myeloma, just not the CAR T, and it are so high that in Belgium, the government has decided that CAR Ts are reimbursed in second line already in multiple myeloma, because it will cost in the end, cut the costs, So in certain fields, the companies will stay in the field of the CAR Ts, but we're not even sure about that, I would say, because there is also the concept of the point of care, where the cost can be reduced by a factor of 3 or 4 or 5.
That is making the products each
a part in the hospital, that you do it, So it's the costs are between 30,000 to 80,000 euro,
for the CAR T cell.
Yes.
Oh, right, exactly.
While for the companies, the cost is usually around 300,000 euros, which is, I can understand that. I mean, I am happy that the companies have gone into the field, that they have convinced the government to do that. But they have also their costs, they are investing into it. They are investing 1 billion euro at least for the development of a drug, and sometimes they have to withdraw the drug even if the first trials were okay. So they cannot do it for everything, and this is what we have for the moment is personalized treatment. So they are pretty reluctant in doing that, In the dendritic cell field, there was the successful and in the end the story of Provenge, where dendritic cell-like cells were shown into clinical trials, to a double-blind randomized clinical trial to have effects for increased survival in prostate cancer. The second trial was done because in the first trial they didn't add the overall survival in the endpoint. So they did it again. They showed it again. It was recognized by the FDA, but then after a while they stopped their operation because of financial constraint. Mm. So in this field, I think, and because this is my past as an hematologist involved with stem cell transplantation, there should be also academic studies, that should have reimbursement. Yeah. If the companies want to join, they are quite welcome, but it should not be completely exclusively in the hands of companies, at least in the field of dendritic cell vaccination.
So are you guys getting to the point where you can file a marketing application to get it fully approved?
Yeah, but we have decided not to do it, because we don't have the resources to do that.
So you'd run it as a permanent IND and then try to get reimbursement?
And then we will restart the negotiations with the government to try to get them, So my successor who has developed the IL-15 dendritic cells, has written a whole item to have the cultures, to have reimbursement per preparation, quality control so much, preparation of the counting of the cells so much, and so on. So like we're doing it for the stem cell transplantation. So after the end, now that the COVID crisis is over, and the person who was involved with the COVID crisis is now in charge of the health, the national health insurance. So we would like to restart the negotiation of that. This is the way, because the marketing authorization, we don't have the money for that. We also don't have the money for phase 3 randomized trials. So we have to convince them that based on the phase 2 studies, if we have clinical activity, which was also in the field of the first CAR T reimbursements in lymphoma were reimbursed on the basis of phase 2 studies, and not on basis of a randomized phase 3 studies.
Yeah, I think that people really don't realize this progression that is necessary, and the differences between having point of care exemptions or hospital exemptions, in which you're only allowed to treat the patients that are in your institution, and oftentimes this is the only way in which you can provide this. It's not self-evident in a way, and
even hospital exemption. This is dependent on the interpretation by the local country regulations. So, for instance, in Spain, hospital exemption is accepted to be broadly used. In Belgium, it's not. And hospital exemption, some people had hoped that it would exempt them from GMP. No, it does not. You still have to comply with the GMP requirements, and you're not allowed to use it as a routine basis, at least in certain countries, including Belgium. So it's not a solution, according to us, it's not a solution, a general solution for these aspects,
Could you go to the regulators and say, hey, we have this phase 2, we want to do a rolling, almost open label phase 3. So for the next 5 to 10 years, everyone we have come in, we're going to collect data and use that as a marketing application. So then you can eventually just get it, approved, but then you're not having this cost problem of I need $1 billion for a trial.
There is I think it's called Article 56 in the Belgian legislation, according to what you can go to the health authorities and ask them that they subsidize, that we get them and that we collect the data, But I've applied for it, I think 15 years ago. I didn't receive that at the time, Yeah.
It's a new day, right?
Yes. So this is another venue that we can get it, But I think in this field, the persistence is the most important thing. And for the moment, the way we've supported ourselves was with the grants and with the and with the charities. What is also happening is that we should be open to new developments, and people are thinking about in vivo targeting by mRNA, So the exosomes, there's different new developments. So this is things about which we are thinking, that we may want to go to, So it's a complex answer, There is no black and white, but you need to have determination to continue working, especially if you have some clinical effects in patients.
Yeah. So I think picking up on that point that you just mentioned, so for the dendritic cell vaccination, you mentioned you're using just naked mRNA. But of course, we all heard about mRNA vaccines, and there's the whole kind of, there's all the whole different possibilities of different types of mRNA molecules. So how did you use non-modified mRNA because that's what was available, or because the immunomodulatory properties of that, the immunogenicity of the mRNA was convenient for activating your dendritic cells? And what do you think is kind of the next steps in using mRNA for vaccines or cancer?
The way we went is because we were not equipped pharmacologically. If you use modified mRNA, this is a whole pharmaceutic operation, with toxicity studies. And remember, there was a talk by Katalin Karikó, who got a Nobel for that, mentioning that in the early days of mRNA development, of the naked mRNA but modified, there were certain toxic compounds, which killed volunteers, because they got trapped in the mitochondria. I remember, not the ones that are being used nowadays, So, this is something that we cannot offer, So what we can do is the production of mRNA, not modified, but with a cap and a poly-A tail, this we can do. There are kits, and this is then documented grade mRNA, and this is not a starting material. So we don't inject that directly into the patient. So this is not a drug. And so what we do is to inject into the patient. And the advantage of mRNA is that if the molecule would have had an untoward effect, mRNA is labile, so it will be broken down after a while. So you're not faced with a potential catastrophe, a regulatory and health catastrophe.
This is instability.
The stability, it's a good aspect. You don't really know if it's good to have these molecules there. And there were the early studies there was with the T cell receptor in melanoma against MAGE, which then the retroviral introduction into in the two lymphocytes which were associated with cerebral and cardiac lethal toxicity, So to have something, a gene being expressed forever in the cells, we were wary of that, So we were very happy with that. But this being said, we were very happy that the companies that Ugur Shahin and Özlem Torici and other people from the company in America decided to go for the modified mRNA, and to encapsulate that in the liposomes. The liposome is also something that we cannot really produce on our own. We can buy that from a company, but this is not clinical grade to be used in patients. So this is outside of our reach. So this is while the mRNA that we produce or that we had produced for us by companies, and the setup of the electroporation in the cells, we do it ourselves. With a regular electroporator, clean. So this is something that we've had. We had the complete freedom to operate there. If we use modified mRNA, there was no way for us to deal with it. And so we would have had, it would have lasted forever and it would not have been started.
Yeah, better done than perfect.
Yeah. Regulation's tough.
Yeah,
but there is a reason for regulations.
Oh, absolutely.
I don't want to give the impression, that this is the regulators are horrible. task is to protect the public, to protect the patients, and you would say, okay, I will work very cleanly, it will be done, but you have to protect the patients from crazy things, and so this is like, and we have to accept that, this is part of our life, and I think it's counterproductive just to go against it, it's not, we cannot, So it's their role, and we have to conform to that, So it's tough, but there's a saying in Dutch, that difficult is also possible, and so it's that's the way we look at talk.
Indeed. So in the last couple minutes, because we just have a couple minutes left, in 2030, where do you want to see dendritic cell vaccines? What's your vision? If you, knowing what the realm of somewhat possible but difficult, what would you like to see?
For the moment, we don't have, according to me, not a real alternative, very easy molecule alternative in vivo. It's not really clear. So I would like it to be able to have point of cares in many places, and that it's being used more, and that also people can start working on improving the system, like it was done for stem cell transplantation at the time, In the beginning, when stem cell transplantation was started, all the patients died of it, and so, but nobody died of a dendritic cell vaccination for the moment, as far as we know. So it's a relatively non-toxic treatment. I would like it to be more accessible and to have more production, And in the meantime, also research to try to develop things that are more are easy molecules, but it has not come very clearly forward right now. People were trying to target dendritic cells in vivo using a receptor in dendritic cells, but this effort, of monoclonal antibodies targeting fusion antibody targeting a receptor on dendritic cells together with the antigen, but it has not, it was very well meaning, but it has not translated into large-scale clinical trials and utilization. So my past as an hematologist makes me believe that, and the same thing, and the confirmation by the CAR T cell field, makes me feel that the cells are still essential in this field,
All right. Well, given that, I think it's right about time for some questions and answers from the audience. So we're going to transition to that here and keep the show going for a little bit. So if there's questions, you can start heading up to the mic.
Good evening, sir. It was a really amazing talk, and your persistence is really admirable. Continuing to the question, I would like to ask the dendritic cell, significance of the dendritic cells in type 1 diabetes.
There are trials of tolerogenicity in diabetes. I'm not sure about the results of that, but I think it has the potential to induce immunological tolerance. Probably the best setting would be at patients at risk, at a very high risk of it developing, or in the very early stages before their islet cells have been destroyed. So this is maybe the setting where it should be
adolescent stage of the
something like that. Someone on the way to, because as there is some immune tolerance treatment. So when those patients receive insulin infusions, there is sometimes of something like an immune honeymoon, where they go in kind of remission, or the period is before they're developing the full-blown disease, So I think there is some stage for it. The antigen that should be used, yes, that's another matter, because
So could there be something like isolating the antigens in type 1 diabetes and clubbing it with the dendritic cells to present and make some antibodies for therapeutic treatment of type 1 diabetes?
I think it's all very good ideas, The important thing is that the clinical trials are the important things, to move forward. I think you cannot tell, and there are some animal models which are encouraging, but unless you go into clinical trials in patients, we won't know for a while. There might also be an indication for immune tolerance if when you do with dendritic cells, when you do islet cell transplantation, and this is another mode, but then the islet transplantation, if they're not encapsulated, then they might be destroyed again by the autoimmune process, So it could be part of the whole treatment. There's a whole field with tolerogenic dendritic cells. Now, this being said, if you ask me to be honest, in the field of autoimmune diseases, the CAR-T against the B lymphocytes have shown some remarkable results, spectacular results, even if in diseases that were supposed to be refractory to everything, like the systemic sclerosis, So I'm not sure, I would love to believe, and we're doing trials in multiple sclerosis, I would love to believe that it's going to work, but whether it's going to be the solution or the whole solution, I'm not sure yet, Yeah.
Hi, it's really cool to be here. I have a question. Do you think that when combining DC vaccination with other immune therapies, that it might get easier to get to that point of a clinical trial and then use the synergistic effects of both therapies?
Easier is a difficult word, but I think we have evidence that it is working. And this has been the idea that has been put forward, that the low level of overall response by cancer vaccination in general could be supplemented by, for instance, the immune checkpoint inhibitors, And we've seen something similar clinically in mesothelioma, where we have had, in the second trial that we've been doing, patients received, untreated patients received dendritic cell vaccination together with the chemotherapy, and then continued to receive the dendritic cell vaccines, and then all of them relapsed, the disease progressed, and then we started giving them immune checkpoint inhibitors. First nivolumab and then afterwards atezolizumab. This was easier said than done because at the time the monoclonal antibodies were not reimbursed by the health insurance. So we had to ask that patient by patient from the companies, would you please give us some monoclonal antibodies, please? And so you find, and then we started seeing things which I think are unprecedented, like a patient in third line who was dying, who then received nivolumab and then went into complete remission, remained in complete remission for 4 years, relapsed of his disease, and then remained in a pretty stable situation and died from something else. Another patient in second line received the immune checkpoint inhibitors, also nivolumab, and went into complete remission and still in complete remission. So I think there is a lot of place in the combination of the two things. And for instance, in other studies, for instance, glioblastoma, where we would, then we have seen the median overall survival is practically double what you have then in when you use chemotherapy alone or when you use the combination of the double checkpoint inhibitors. So I'm convinced that it will help. But to get those immune checkpoint inhibitors, that's another matter. And we have had contacts with Uğur Şahin and Özlem Türeci, the developers of the COVID vaccine, of Pfizer BioNTech vaccine. And of course, their start was in the cancer field, and then they wanted to get away. And I am glad I'm glad for science and for medicine that they decided to go to the generic way of the mRNA alone, so that it's accessible to more patients. And they are doing the clinical trial. They have clinical trials with Claudin mRNA. But they use also monoclonal antibodies. And I talked to them, how do you get them? I said, we have to buy them for every trial, for every patient, so we have to buy those products. And if we wanted to buy that in the clinical trials that we're doing, the funding agency will not give the money for this month to buy these checkpoint inhibitors, because they say we are not going to give money to these rich companies, we're not going to give them money for that. So easier conceptually, certainly. I believe it's going to work. The other way is then to try to get that, So I think this is one way of going, and we have evidence for that and it's not published yet, for the mesothelioma. The concept of having more specific effect against the tumor-associated antigens with the cancer vaccination, supplemented with the immune checkpoint inhibitors, which are more more general. Also, you could supplement it with chemotherapy, actually, and we've heard about that. Laurence Itfogel, who done such a good job on the microbiome in her past. She has introduced the idea of the immunogenic chemotherapy. So chemotherapy is reducing the blood cells, like the neutrophils and the red blood cells, but it has certain chemotherapeutic agents have a T-cell stimulatory activity. So the combination of the two also in our hands has probably some additional effects, So I think this is the way to go, to try to increase the effects of the cancer vaccination.
Well, thank you.
Okay.
We have time for one more question.
Thank you for this brief history on dendritic cell-based immunotherapies. I was wondering whether you have any idea why dendritic cell-based immunotherapies for cancer only work in a number of patients, and have there been any efforts to get behind that?
Well, I think it's similar to what we see with the immune checkpoint inhibitors, and it's the same thing. It doesn't work in all the patients. And similar also in stem cell transplantation, acute myeloid leukemia, when we even transplant the patients in complete remission, part of them, a quarter, will relapse of the disease. And the reason is probably the T-cell stimulatory activity is not strong enough in that case. And that it could be due to the fact that the dendritic cells or the cancer vaccination is not strong enough, or maybe that the T-cells are not there anymore. They have withered, due to the cancer. So those are, I think, the main reasons. When I talked to my colleague in Japan who is doing Haruo Sugiyama from Osaka, Japan, with the W2 and peptide vaccination, his vision is that it works in all the cases, and that, for instance, the glioblastoma patients who are doing horribly without the vaccination, they will then do badly. So the curve is the survival curve is switched to the right, to the right. So the very bad patients are doing bad. The intermediately bad patients are doing moderately. The better prognosis patients are doing better, So you're just shifting the curve, This was his idea, that it's working. And we have some evidence for that, because even in the failures of immunotherapy in general, I'm talking about, for instance, the Provenge vaccines, there was a sub-study done there that the patients who failed the vaccine, so who relapsed after the vaccine, then received chemotherapy, they had a longer survival than the patients who did not receive the vaccine first, So even maybe the failures of immunotherapy may not be complete failures, and that they're preparing the field for there, So it's, and there are some evidence in other fields, So it's not complete success. We don't have 100% in all the fields of medicine, But I think it's due to the fact that the T-cell stimulatory capacity is not optimal. I think this, and it could be due to different reasons,
and also depending on disease state and then the state of the cells,
yes,
which treatments they got.
Yes, absolutely. Yeah.
Thanks for those insights.
Yeah.
All right. Well, that brings us to the end of the live show. We'll be here afterwards for extra questions. But first, thank you, Dr. Berneman, for coming. Thank you, IUIS, for hosting us here. This episode comes out August 26th. You can find it at immunologypodcast.com or wherever you get it. If you want to reach out to us, it's info@immunologypodcast.com, on Twitter, on X, at Immunopodcast, and then at Blue Sky at immunologypodcast.com. That's it.